Okay. I will be very brief, not only to save time for the discussion after Peter Raven's presentation, but because I came here not to talk. But Marcelo convinced me that I should talk, and so I prepared 6 slides yesterday evening to give you a message. And I borrowed 1 slide from Taylor Hensch, which you have seen in his presentation, because I like this slide, which illustrates beautifully the difference between curiosity-driven research and directed research or targeted research. I'm sure most of you have much of sympathy for this little chick in front, because you are all, as far as I understand, proud of doing basic research. I must confess I am this fat chicken. Since my beginning of my academic career, I had a very specific target, and all what I have done has followed this target. So forgive me if I'm talking to you from this perspective. I was actually attracted to do science by the phenomenon of totipotency of plant cells. I have been teacher in high school for 10 years. When I had the chance to do a PhD in the Max Planck Institute for Plant Breeding Research in Cologne, and my thinking was influenced by this time at the Max Planck Institute. And I was concerned about food security already in the middle of the 60s. I have been teaching special courses to my students on the problem of food security already at that time. So when I had a chance to enter a career in science, I was somehow strongly influenced by the wish to contribute to food security and by the unique phenomenon of somatic plant cells to have the capacity to regenerate to complete normal plants if given the right conditions. The concept of totipotency has a long history. It started with the concept of Schleiden and Schwann, that cells are the basic unit of multicellular organisms. Over Haberland, who hypothesized that plant cells must have some kind, the potency of regenerating to plants, because tissue culture experience showed that there is such a potency. Then it continued with the first successful establishment of callus cultures, which was the starting point of a very successful area of plant science, tissue culture. This was followed by meristem culture. Meristems are the tiny growing centers of plants by which they build up the entire plant body and the roots. These are specialized multicellular structures. This was followed by the discovery of plant growth regulators, which play a role in this effect, followed by the establishment of root cultures, which means that wide, isolated, under sterile conditions, roots, and took them into culture, and these roots proliferated and made more and more roots. White, Gauteret, and Nobelcore established unlimited callus cultures from plant material, which indicated that plant cells are probably have an unlimited capacity to proliferate. You can see in trees which are 3,000 years old, and you can still isolate meristems from these trees, and they still continue to grow. This was followed by embryo cultures, the culture of isolated immature embryos in synthetic media, followed by shoot cultures, the successful proliferation of shoots from shoot meristems, followed by suspension cultures. Suspension cultures are comparable to bacterial cultures because they consist of single or small groups of cells which are cultured in flasks on shakers and obviously have the capacity to grow unlimited. Then came the discovery of two important plant hormones which play a role in the differentiation and development of plants as well as in tissue culture. Then came the first important example if you consider the question whether single cells are totipotent. Bergman isolated single cells from suspension cultures and showed that single cells have the capacity to proliferate. Murashige and Skoog designed a totally synthetic cell culture medium. In plant cell culture, you do not need complex and undefined extracts and additions. You can work on the basis of a totally, fully reproducible and synthetic culture medium, followed by somatic embryogenesis from carrot suspension cultures, followed by embryogenesis from mechanically isolated single cells in carrot, and finally, to everybody's surprise, the fact that you can regenerate haploid plants from structures which are comparable to male and female gametes. So there is obviously an enormous plasticity in the capacity of plant cells, somatic plant cells, and the coin totipotency was coined in 1968 by Steward. Cocking added another aspect by isolating naked plant cells which had been stripped off their cell wall. Even these naked plant cells have the capacity to proliferate. And Nagata Takibe were the first to demonstrate that you can establish callus cultures, which means undifferentiated proliferating plant cells from naked protoplasts. And this was the time when I joined science. And you see here, my first contribution to this development was the report on the recovery of fertile total plants from Petunia protoplasts. At that time, this was so exciting that it was a Nature publication. So, I entered science with a specific idea. I was interested in totipotency, not because I wanted to find out how totipotency comes about in plant cells, but because I wanted to use it for a specific purpose. And the specific purpose I was trying to achieve was to develop plant breeding technology on the basis of large populations of single somatic cells as a complementation to existing plant breeding strategies to contribute to food security. What means large populations of single somatic cells? You must imagine that 1 gram of leaf tissue, 1 gram of leaf tissue contains more than 2 million cells. These 2 million cells from 1 gram of plant tissue can be isolated, taken into culture, and have the capacity to regenerate to plants. For somebody with a plant breeding background, this was irresistible to challenge. And as the idea was to change the genetic makeup of these somatic cells, we spent a few years in trying what is all possible with these naked plant cells. Before I come to this, let me emphasize to you that when I started this type of work, it was to everybody's understanding firmly established that living plant cells are totipotent. And this was the basis for what I was trying to do. Well, as I indicated, I spent about 4 years as a little chicken playing around, trying all different things which were possible, fusing protoplasts to establish somatic hybrids, fusing subprotoplasts with nucleoplasts, transferring organelles into protoplasts, transferring DNA, and so on. This was a lot of fun, but it was not on the line of my intentions. My intentions were to contribute to food security, and for this purpose, I had to work with plants which are important for food security. So after 4 years of fun, I decided, now I do serious work with cereals. And I thought, with my experience, it should be not too difficult to regenerate plants from wheat protoplasts. Well, you see here, 1971 to 1986, attempts to find culture conditions enabling cereal mesophyll protoplasts to realize their potential. Mesophyll protoplasts are naked cells from leaf cells. And this was considered a basis for cereal breeding with large populations of these wheat cells. Well, I invested 1 intensive year of work of cell culture, and the response from these wheat cells was nothing. I invested a 2nd year, and the response was 0. I invested a 3rd year, and the response was 0. I invested a 4th year, and as you see here, I invested altogether 15 years of very hard and very intensive experimental work done by myself. I did not dare to put any student or PhD student on such a risky program. So 15 years of my life I invested into tissue culture of wheat mesophyll protoplasts, applying everything which ever had been successfully or unsuccessfully applied to tissue culture problems. And after these 15 years, I had, if you believe me or not, tested more than 120,000 different variations of tissue culture treatments, going up to 7-factor gradient combinations of all possible factors having been tested. So after 15 years, I asked myself, how long can you continue without looking stupid? And I decided I give up. I gave up with the assumption that obviously not all plant cells are totipotent, but only some plant cells for which it has been demonstrated. And obviously, or possibly, differentiated cereal cells are not totipotent. We don't know yet, and deep in my heart, I'm convinced that if I would have 10 more years and experimental support, I could convince wheat mesophyll protoplasts to regenerate to plants. So I gave up because I felt I had spent enough time on this. So I had to change my concept of totipotency and modified it in that way that I now believe that totipotency is a character of somatic plant cells for which it has been documented. And there are incredible documents like plant regeneration from needles of fir trees or from many other trees. And as I said earlier, from virtually every single plant organ. So what do you do in such a situation? If you would be a curiosity-oriented scientist, you would try to find out what's wrong. What is the difference in regulation of the somatic cells in herbaceous dicots compared to gramineous monocots? This would have been a scientific project, and I'm happy that I did not follow this line, because until now we don't know. Many colleagues tried to find out about the difference. Many colleagues tried to find out more about the phenomenon of totipotency, how it is regulated, what's the difference. We have virtually the same level of knowledge as we had 40 years ago. So I needed a way out of this roadblock. And the way out of this roadblock was to prevent differentiation and to multiply serial cells under conditions which allow proliferation but prevent differentiation. And this was the beginning of embryogenic suspension cultures. And these embryogenic suspension cultures allow what we wanted to do, to build up large populations of somatic plant cells with the competence for regeneration. And from these embryogenic suspensions, you can isolate protoplasts and introduce DNA, and finally produce golden rice. This was the way how we finally ended up with golden rice. So we achieved our goal of developing technology which contributes to plant breeding. You can apply other gene transfer technologies to this system as well. And if you hear and see about any transgenic cereal nowadays, that's the experimental basis. And this is what I wanted to tell you. I understood the invitation to contribute to this symposium, that we give an example of how a concept of nature had to be changed as a consequence of experimentation, and you have seen this example. And I also wanted to show you that so much I value the importance of basic research. If I wouldn't have followed this targeted way, we would not have golden rice. There is no question. We would not have golden rice. We would not have many other achievements. My concept of a science is that we are responsible for doing excellent research, but we also have some social responsibility. And that's how I try to follow both requests. Thank you for your attention.